Hi, my name is Rowan Wooldridge. I am a biochemistry major and I'm studying methyl-accepting chemotaxis proteins in sinirhizombium meliloti. So S. meliloti is a symbiont of known rotation crop alfalfa, and alfalfa will release these molecules into the soil in order to attract S. meliloti to the site of its roots, and it will provide the bacteria with all of the nutrients that they need to grow, and in return, S. meliloti will fix atmospheric nitrogen that the plant cannot obtain on its own. The targeted movement of S. meliloti in the response to a specific molecule is a process known as chemotaxis. These bacteria possess sensor molecules called methyl-accepting chemotaxis proteins, or MCPs, that bind to the molecules that alfalfa releases and causes the bacteria to swim towards them. My job in this project was to isolate 2 of the 8 MCPs present in these bacteria and determine which molecules they sense. This is important because if this process were to be optimized, it would result in soil being naturally enriched with nitrogen and thus reducing the need for harmful chemical fertilizers. To start this process, I first cloned the genes coding for both of my proteins of interest into 4 different vectors each. I then tested to see which of these constructs within E. coli would produce my protein in a way that would allow me to purify it. This gel here compares 2 different constructs, the genes coding for McpW within PTYB1 and PTYB11, and as you can see, there is no real difference between the uninduced and induced wells here, but for this one, there is a clear band in the induced well that is not present in the uninduced well, which means that the protein was expressed. Similarly, there is a band at the same molecular weight in the soluble fraction, which means that we can collect this group of proteins and move on to purification. This table over here describes all 8 different constructs and which ones worked and which ones didn't. I started by purifying McpW using impact affinity chromatography, but the soluble protein concentrations never were high enough to analyze the protein in the next step. I then moved on to try to purify the other protein McpZ using the same impact affinity column chromatography methods followed by size exclusion chromatography. This gel here, you can already see that there is a very good amount of the pure protein. The chromatogram to the right of this gel also shows the purified McpZ after size exclusion in very good amounts. The next step was to determine which ligands bind to this protein using differential scanning fluorometry, or DSF. This process was repeated several times with several hundred different potential ligands, and no compounds were determined to bind to McpZ. So we decided that it was possible that McpZ was binding indirectly to the ligands, and therefore DSF really wasn't going to help us characterize them. Simultaneously, we are collaborating with another lab in order to crystallize this protein to give us an idea of its structure. This will give us a better idea of what kind of ligand could bind to McpZ. This picture in the top right shows the preliminary crystals of McpZ, but this process is still very much underway. Another method I am using to determine what compounds McpZ could detect is a capillary assay. This process is outlined in the schematic here, but basically you put 1 microliter of a potential ligand inside of a capillary and place that inside of a culture of S. meliloti. And if the ligand that you put in the capillary is an attractant, then a lot of the bacteria are going to swim in it this way, and once you plate it, you can count how many colonies form. So the stronger the attractant, the more bacteria will swim into the capillary and the more colonies you'll have on your plate. And so basically you repeat this process for a deletion strain of sinirhizombium meliloti that does not contain the genes coding for your protein of interest. And if the wild type did have a lot of bacteria swim into it, but the deletion strain didn't, then that means that your protein is implicated in sensing that molecule that you put in the capillary. So this process is definitely very much underway too. I have already tested a few compounds and haven't gotten a super strong attraction to any of them, but I'm still very much working on this. Once again, this project is being done in hopes that the symbiotic relationship between alfalfa and S. meliloti can be optimized, leading to the reduction of the use of harmful fertilizers. Thank you guys for listening, and let me know if you have any questions.